Okay, we're going to get back into our genetics lectures here. And in the last video, we looked a little bit more at Gregor Mendel and some of his discoveries that he came up with while he was doing his experiments with those pea plants. Now we're going to take that a step further. And we're going to use his ideas and we're going to come up with our own discoveries. How do we determine crosses? What happens if you have a tall pea plant and a short pea plant? How do we figure out what are the potential outcomes of that cross? And more realistically, we would use that in, if I have Tay-Sachs disease in my family and my wife has Tay-Sachs disease in her family, what are the chances of us having a kid with Tay-Sachs disease? Well, that's obviously something that you'd want to know if you're deciding you want to have children. So we're going to use something called probability. Now, you've heard that term before. Probability is basically we're just looking at potential outcomes of a situation. If I took a coin that has a heads on one side and tails on the other, and I flip it 100 times, you probably know this answer already, but how many heads would I get? Well, I don't know for sure, but chances are I would get around 50 heads and 50 tails, right? Because there's only two options. And when there's two options, every time you do that, whatever it is, flipping a coin, there's potential of getting either one equally. Now, the same thing is true actually with having children in terms of a male or a female. You have a 50% chance of having a boy or a girl every time a baby is conceived. And that might seem odd because you might have friends that, maybe you have a friend or maybe even your own family where you have two brothers and you're a boy too, or you have two sisters and you're a girl and you've only had girls in your family. Well, you can flip a coin 100 times and you can get 100 heads. It doesn't mean that you're going to get 50-50. It's just that based on the rules of probability, chances are you're going to get 50 and 50. The same thing is true with boys versus girls, right? So if you take a look here, this is called a Punnett square. And we're going to talk about Punnett squares a little bit today. A Punnett square can be a little bit intimidating. You probably learned about this in middle school. And you probably memorized how to fill it in. And you probably spent some time with them. But maybe you didn't think about why, right? And all a Punnett square is, it's a tool, right? If I gave you a nail and I said nail this into a piece of wood, and you looked around and you had a shoe and you had a hammer, which would you use to nail that, nail into the piece of wood? You probably would use the hammer, right? Could you nail it in with the shoe? Yeah, you probably could. If you sat there long enough and hit it hard enough with your shoe, you probably can nail that into the wood. But guess what? You're wasting your time and your energy when they already invented this thing called a hammer, which is sitting right there. So think of a Punnett square as a hammer. It's a tool to use that makes it easier to figure out the outcomes of a cross between different individuals or whatever we're talking about. So if you look here, this is a Punnett square, and we're looking at up here you can see two X's, so that represents a female, and here is an XY, so that represents a male. And I'm going to go into more detail on this in a moment, but all a Punnett square is it's a way to determine what the outcome of these crosses could be. Now, what are the chances of getting boy versus a girl? 50-50, right? Now you might be looking here and saying, well, wait a second, Mr. Adolf, there's three X's and only one Y. So wouldn't it be easier to get a girl than a boy if there's only one Y and three X's? Well, let me show you why that's not true, right? So this would be one individual here, the two alleles, one X and one X for the sex chromosome. So for the property of sex, male versus female, female has XX, male has XY. So the way a Punnett square works is actually very simple. You're basically going to pass these alleles into the boxes that are next to the allele. So this would look something like this. This X would go into this box and this box. This X would go into this box and this box. This X would go into this box and that box. And this Y would go into that box and that box. Now again, I'm going to go into that in more detail in a little bit. But for right now, we're just going to focus on this. And what you'll see here is there's four boxes, right? And if I look at those four boxes, these two have two X's, so they would be female, and these two have XY, so they would be male. So with there's four boxes, each box represents 25%, right? Because four is quarters, that's 25%. So two 25% are going to be XX or female, and two 25% are going to be XY, so it's going to be 50-50. So even though there's only one Y chromosome, it only takes a Y to be a male. You don't need to have two Ys to be a male. You just need one Y. So male, female is going to be XX. I'm sorry, female is going to be XX, male is going to be XY. Now let's talk a little bit more about why this is a helpful tool. So let's talk a little bit about probability a little further. So here we have heads and tails, right? So let's say I have a coin and I have an H and a T, right? So I have just an H and I have a T, just one coin. And I flip that one coin. What are the chances of getting heads versus tails? Well, we already know it's 50%, right? Every single time you do it. But now let's say I have 2 coins. Let's say I took 2 quarters and I flipped them both at the same time. And I want to know what the outcome could possibly be. Well, there's 4 possibilities that I can get with 2 coins, right? I can get both heads. I can get one tails and the other one heads. I can get one heads and the other one tails, or I can get 2 tails, right? Those are only, there's no other possibility, right? So think about it for a second. Is there any other thing I can get if I have 2 coins? Hopefully the answer to that question is no, right? There's not. So what are the possible outcomes of these percentages? Well, if there's 4 possible outcomes, one thing I want to point out to you is there's no difference between these 2, right? So if I take a look at these 2 here, they're exactly the same. Tails, heads, heads, tails. So if those are the same, we're going to group them together. So what's the percentage chance of getting tails, heads, and heads, tails? That's going to be 50%, right? Sorry, my writing's sloppy. I'm trying to do this with a mouse pad, and it's not that easy. What are the chances of getting heads, heads? That would be 25%. And what are the chances of getting tails, tails? That would also be 25%. So if I have a heads and a tails, if I have 2 coins and I want to see all those possible outcomes, what I would find is that I have a 50% chance of getting tails and heads and a 25% chance of getting both heads or both tails. Now, does that mean if I do this 100 times, that's my numbers are going to be 25, 50, 25? No, but if I do this, I would find that it would be very close to those numbers. So that's just a way to think of this. Now, why do we use a Punnett square? Well, let's take a look over here at my little scenario over here. So let's say I have 2 couples, right? I have a red couple and a green couple. And I want to know all the possible combinations of these couples. Now, I don't want these 2 can't go with these 2. I want to know all the possible combinations though of these together. So the first thing I could have is I could have a red and a green, right? I could have something that looks like, oh, sorry, I just said I can't do that. I could have this, right? A red can go with the green in this scenario. A red can go with a big green. Little red can go with a big green. Little red can go with a little green. So I just basically looked at all the possible combinations. Now, that was kind of annoying to do, right? So what if I did this a little bit different and I put them over here with the Punnett square? Now, I just have a much easier way to organize this stuff. This red guy would go here, and this red guy would go here, right? Because it's lined up with that row. This green guy would go here, and this green guy would go here because it's lined up that way. This little guy would go here, and this little guy would go here because it's lined up that way. And then finally, this little guy would go here, and this little green guy would go here. Now, what I just did was the same exact thing that I did over here. It's just this is a lot more organized. And as you can see, these are my 4 possible outcomes of a cross between these 2. So we're going to use that idea, we're going to go through some examples, but I just wanted to explain to you why we use this idea. Okay, so let's jump back into the notes, and then if you guys have any questions, you can email me. Okay, a couple more terms, and then we're going to finish up for today. Heterozygous versus homozygous. Now, you probably remember this before. Hetero means different, homo means same. You can remember that pretty easily. You know those terms, right? Someone who's heterosexual, like somebody who's the opposite sex, So male, female, or however you want to think of that. And someone who's homosexual, like the same sex. So homo just means same, hetero means different. So when we're looking at our alleles here, basically there's 3 possible outcomes that you can get from 2 alleles. You can have homozygous dominant. Zygote refers to chromosomes or genes, right? So homozygous dominant means that both alleles are dominant. So in the case here, it's uppercase. So big T would be homozygous dominant, and that's always going to express the dominant trait. So big is always going to give you the dominant trait. So if we're talking about tall versus short, where tall is dominant to short, it's going to come up tall. So homo means same, dominant means big T. Homozygous recessive, homo means same, but now recessive is going to be the smaller letter, right? So 2 identical recessive alleles that are lowercase. So that's going to be little t, and that's going to be short, okay? And then finally, we have heterozygous. So heterozygous means it has 2 different alleles for the same trait. So it's going to be uppercase and lowercase. Now, here's the thing with alleles. you always want to put the uppercase letter first. So if it was little t, big T, that would be written wrong. So it's always going to be the big letter first. Second letter is going to be the smaller letter of the two. So big T over little t. And in this case, for a dominant versus recessive, if it's complete dominance, which we'll talk about later, the dominant trait is going to be shown in this scenario. So heterozygous is always going to be the dominant trait. So just as a review, you have homozygous dominant, which would be 2 big letters, homozygous recessive, which would be 2 little letters, and then heterozygous, which is going to be big letter, little letter. And that's going to represent the dominant trait. Now, one more term I want to go over with you guys today, or 2 terms, and then we'll be done for today's lesson, is genotype versus phenotype. And this is one that gets a little bit confusing, so I want to take a couple minutes with this. So the genotype refers to the actual genetic makeup. This is the code in your DNA. So for example, if we're talking about height here, the code would be big T, or little t, or big T, big T. That is going to be the genotype. So that is what's actually in your DNA. The phenotype is the physical expression of that trait. So big T would be tall, where little t would be short. So if you look at this little diagram over here, this is a good example. So this is going to be the genotype. So there's 4 possible genotypes. You can have big P, you can have big P, you can have little p, big P, which is the same thing. That's why they're written twice. Or you could have little p. All 4 of these options are genotype. That is the actual code in the DNA of this plant. Now the phenotype is what is it actually going to look like. So in this case, purple is going to be dominant to white. So big P is going to be purple, right? Because the dominant is going to overpower. There's no recessive at all here, actually. These 2 are both going to be purple because big P overpowers little p, making it purple. And then finally, little p is going to be white. And that is going to be both recessive alleles, which is going to give you the recessive trait. The only way you're going to ever see the recessive trait in a complete dominance problem is if it's 2 small letters. If it's homozygous recessive, that's the only way you're going to see that in this trait. Okay, so I'm going to just quickly review what we talked about today, and then we're going to actually get into doing some problems in the next video. So I don't want to jump into that just yet. So quick review what we talked about today. We talked about the idea of Punnett squares and using them to determine the outcomes of crosses. We talked about the fact that there are 3 possible, I'm going to back up here, right? There's some possible genotypes here. You could have homozygous dominant, which would be big T, both uppercase letters. You can have homozygous recessive, which would be 2 identical recessive alleles, or little t, or you could have heterozygous, which is big T, or it does the letters don't matter. It could be any scenario. It just means big, little, dominant, and recessive, and that is going to represent the dominant trait. And then finally, we talked about this idea of genotype versus phenotype. Genotype is going to be the actual DNA. For example, in my DNA, it codes for male pattern baldness. Now, you can't see that. That's the genotype. That's what's in my DNA. But if you look at my head, you can see that I have male pattern baldness. That is the phenotype. So the physical expression of the trait. So my DNA codes for blue eyes. My eyes are actually blue. So the DNA coding for my blue eyes is genotype. My actual eyes being blue when you look at me is the phenotype. Okay, thanks very much. Hope you guys have a good day.